Good morning, good afternoon, maybe for some folks. Thank you again for joining. As you all know, this is the second lecture as part of the UCLA CERM and Thermo Fisher training around pluripotent stem cells and their use in the lab, as well as in later discussions, we'll also talk about potential use in the clinic. But for today, and I should take one step back. I should reintroduce myself. For those of you who weren't able to join us on the first call, my name is Omar Farah. I am a field application scientist at Thermo Fisher Scientific, and I'm joined by my colleague, Tom Forbes, who's also a field application scientist. We both support the pluripotent stem cell and neurobiology workflows under the greater cell biology umbrella at Thermo Fisher. And we're happy to kind of give you guys a little bit of training, a little bit of a deep dive today into reprogramming techniques and tools. And Tom mentioned this last time. I will re-emphasize it. If you have any questions or have any comments, please feel free to come off mute, drop something in the chat. We want this to be as interactive as possible. There's no bad questions, no stupid questions. So please, if there's anything, let me know, let Tom know, and we're definitely happy to help. Okay, see if I can move these slides along. There we go. So for today, I'm going to be giving you a brief introduction into reprogramming, giving you a little bit of background and talking about the Yamanaka and reprogramming factors. We're then going to do a slightly deeper dive into the different reprogramming methods and technologies. We're going to focus specifically on the cytotune Sendai reprogramming, which is one of those reprogramming methods as it's one of the more widely used methods as well. We're going to talk about different workflow and media considerations as well. And then finally, towards the end, what I want to do is start to introduce the concept of PSC characterization and what basically that is and why we do it. And uh, there we go. Just as a quick recap from last week's lecture that Tom gave, we kind of started talking about what pluripotent stem cells are, where they originate from, right? In vivo from typically from the embryo at the blastocyst stage, and what their potential applications are when it comes to basic biology, drug discovery models, and cell therapy. But Tom did go over a brief example of disease modeling, where where you can use these cells in order to make a genetic mutation in a very specific gene that we know is related to a certain disease. Last week, we gave an example related to Parkinson's disease, where we could then create two different cell lines, one with that mutation that resembles a disease, and then one that would be wild type, and then compare the outcomes of when we differentiate these two lines into a very specific type of neurons that we know is lost during Parkinson's disease, that being dopaminergic neurons specifically. So I hope that gave you an idea about how we can use these cells in the lab and sort of what things we're going to be talking about throughout our entire lecture series. And you'll notice from this point forward that our lectures will follow a very similar um, uh, pattern to how we use these cells in the lab, right? Typically, we describe that as a workflow, right? How do we work with these cells from start to finish? And so looking at the very left side of the slide over here, you'll see that most folks will start with a somatic cell where they can culture that cell, whether that be fibroblasts or blood-derived cells. And then they'll move over into what we call, and we'll talk about this today, reprogramming, where they can reverse time, if you will, and take that somatic, fully differentiated cell back into a pluripotent state. At that point, you would have created a pluripotent stem cell line, and you go on to culture that line, characterize it. If you want, then you can move to engineer that line, genomically change specific genes or loci, depending on what you want to study, before you differentiate these cells into whatever subtype that whatever cell type you're, again, looking to investigate. and doing your experiments and measurements at that point. So today I'm going to be focusing, like I said, mainly on reprogramming, which is towards the front end of that workflow. And so if I were to ask folks how many folks have heard of reprogramming or know what that is? Drop it in the chat or speak up if you wanted to. There you go. Catherine definitely has heard of it. I know Kita has also definitely heard of it. Tom and I have heard of it. It's okay if you haven't. We're going to be talking about this today. But basically, what reprogramming is it's a process of reverting a somatic cell or an adult cell into a pluripotent state so that it behaves more like the cells at the very early embryo stages, right? And these cells, like Tom discussed last time, these pluripotent cells should have similar qualities. And a lot of this work was actually pioneered by a Japanese researcher called Shinya Yamanaka. And Shinya, alongside with actually some work here in Wisconsin by Jamie Thompson's group, together kind of figured out that you can take somatic cells, right? Typically fibroblasts or blood-derived cells, and if you force the expression of just four factors, right? OCT4, SOX2, CMYC, and KLF4, if you force the expression of these transcription factors, and I should note that these transcription factors are typically expressed in that pluripotent state during early embryogenesis. Uh, these somatic cells, these fibroblasts, if you will start a signaling cascade by which they can reshape the identity of that in particular cell. And towards the end of the reprogramming, if you will, you're left with a cell population that resembles those early embryonic stem cell counterparts. And because of that, we actually refer to these cells as induced pluripotent stem cells, or iPSCs. Notice that the induced here is referencing the forced expression of these four factors in that somatic cell in order to get it to that pluripotent-like state, i.e., what we call induced pluripotent stem cells. And I should note, and we'll talk about this a little bit later on, but due to his work on reprogramming, Shinya Yamanaka actually received the Nobel Prize in 2012. With a lot of us in the field vividly remembering that period where I specifically remember where I was when I read Shinya's paper. I was in the lab in Montreal at the Royal Victoria Hospital. It was a little bit later at night. I was waiting for one of my gels, and I was online. At the time, we didn't even have Wi-Fi in the lab, but that's not because that was our lab mainly. The walls were too thick for Wi-Fi signal to go through. We were in a slightly older lab, but I was on my desktop, and it was so exciting because this discovery really changed our field completely, I would say. And we'll talk a little bit more about this today. So, at its essence, the reprogramming is basically, if you can get cells or dividing proliferative cells mainly to express those four reprogramming factors, or as they're more commonly also called Yamanaka factors, you can get the cells to start that signaling cascade process by which they can become induced pluripotent stem cells. And so the biggest question here is how do you get those transgenes to be expressed in the cells, right? So there's different what we call delivery methods by which you can have constructs or transgenic constructs that you can deliver into the cell and get those cells to start expressing these transcription factors. The main distinction between the delivery methods is whether they're integrative or not, I would say. So what does that really mean? Um, when we refer to an integrative delivery method, this is a method by which the payload that you're delivering, in our case, the Yamanaka factors, get integrated into the genome of the host cell, right? Um, Integration is usually somewhat of a random process with a lot of those delivery methods. Sometimes it's not. But you can see how that could be somewhat problematic, right? If you're trying to force the expression of four factors and in doing so, you end up inserting the construct into a part of the genome that's really essential, right? For example, a proto-oncogene or a tumor suppressor. You can actually, really change the identity of those cells and not really have the same starting genetic background that you did compared to your somatic cell. Now, on the other hand, non-integrative methods are methods by which you can deliver your payloads, right? You can see actually over here. You can deliver your payloads into the cells, but without having that transgene be incorporated into the genome of the host cell. So this is more of a transient delivery method where you get cells to basically act like factories and express your construct. In our case, we want them to express the four transcription factors, right? The Yamanaka factors. But then after these cells reprogram, you don't need that construct to stay in the cell, and we call that vector clearing, right? So at some point, we want to make sure that those vectors are no longer expressed in the cell. And I know this is a lot of words and might be a little confusing. This is a very complex slide, right? But don't worry, we're going to go through this together. What I want to highlight are two very common ways by two very common non-integrative delivery reprogramming methods that we commonly use in the lab. The first is using the Sendai virus, where those reprogramming factors or transcription factors are packaged into a Sendai virus particle, and then those virus particles are basically going to infect your cells. And we'll talk more about the Sendai specifically, but basically the Sendai virus, its payload doesn't, or its payload is only expressed in the cytoplasm of the cell. So it doesn't go into the nucleus, and there's very little chance of integration into the host genome. The second non-integrative method that you'll see very commonly is just delivery of episomal vectors, where you have your transcription factors in plasmids, and then you deliver those plasmids to the cells, allowing the cells to again express your gene of interest, and in this case, as our reprogramming factors. And after a while, you'll see that those plasmids will be removed from the cells gradually as well. Now, um, you see that there's multiple different methods by which you can reprogram, right? And there is definitely certain considerations that we should take that will help us figure out what would be the most appropriate methods for our needs. So when looking at all these different methods, how do you really know which method you want to use? And I put up a bunch of considerations here that are important to highlight. The first being safety, right? The safety profile associated with that reprogramming method. Um, as many of you know, whether and I've already kind of alluded to this with the non-integrative and integrative methods. Uh, the way in which you target these cells to express your reprogramming factors could potentially change their genomic DNA, right? If it's an integrative method. So you'll see that, for example, retroviruses tend to have a higher aneuploidy rates after reprogramming compared to mRNA or Sendai, for example. On the episomal vectors, even though it's a little bit lower than the retro, and it is technically non-integrative, there's been slightly higher aneuploidy rates also noted with the use of episomal vectors specifically. So the safety of the method is important, but also another consideration is the consistency, right? If you have to ask yourself, how many experiments do I have to do to generate an iPS clone with each reprogramming method? And you'll see that for the most part, most reprogramming methods are robust. You see the Sendai virus, the episomal, lenti tend to have really high success rates, meaning that you are able to generate an iPSC clone from your starting experiment. Where we have slightly lower rates is on the mRNA front. And we'll talk more about this specifically, but if you were to deliver mRNA into your cells that encodes the four transcription factors, the half-life of the mRNA is usually a big consideration, right? How long can I get these cells to maintain that initial mRNA construct that I delivered? And is that enough time to get the cells to reprogram, right? And this is a slightly tricky area that we'll discuss, that we'll do a little, we'll talk a little bit more about later on. The third thing we consider usually is the workload associated with the different reprogramming methods, right? How much time or hands-on time or handling time do I need to be as a researcher allocating to when it comes to the different reprogramming methods? And you'll notice here, for example, that the mRNA method again tends to have a slightly higher handling time, and because of the protocol in which we mainly deliver these mRNAs into the cells. But this is really, really important, specifically if you're working with multiple cell lines, right? If you're doing a bigger project where you're creating some sort of bank, right, and you're collecting a lot of patient samples and reprogramming them into induced pluripotent stem cells, do you prefer a method that is potentially a little bit safer and but can take a lot more time, or something that like the Sendai, for example, that is relatively safe but has a shorter handling times, but also maybe associated with a viral component that's not very desirable, right? The last thing that I'll mention in terms of consideration is what is the somatic cell you are trying to reprogram, right? This is important because certain somatic cell types are more suitable for certain reprogramming methods. And I'll give one example is, for example, with blood-derived cells, and if you were to take a blood draw, in order for you to
Yeah.
use an mRNA reprogramming method, you actually have to first take these cells that from blood and differentiate them into endothelial progenitors
Yeah.
so that they become adherent. And this process takes 2 to 3 weeks, right? So this is 2 to 3 weeks prior to even starting your reprogramming experiment that you need to kind of consider, right? So the somatic cell type that you're intending to reprogram is also very important. Now, if we were to place all these methods and consider them based on two main factors, right? Their safety and efficiency, you'll see that we have a quite wide distribution across those two factors for the different reprogramming methods or agents. But what we really want to look for is methods that have really high safety as well as high efficiency. So this upper right quadrant right here, you'll see that the cytotune, episomal vectors, and mRNA are tend to stay in this quadrant as well. Now you see the three methods that are highlighted in red over here are reprogramming methods that we have ready-to-made solutions for at Thermo Fisher, but there's a lot of other ways in which you can reprogram. For example, with the mRNA method. Now, before we start talking about reprogramming in general, we kind of alluded to this. We start this process from a somatic cell, right? And you'll see that the most common somatic cell types that are used for reprogramming are either skin cells, where we take a small biopsy, or from blood draws,
I would say the exact.
where we take blood-derived cells, and we can reprogram some of the cells, either the PBMCs or the CD34 positive cells. These are the hematopoietic stem cell population in the blood, or even other lymphocytes like T cells or B cells as well. There are other alternative cell sources that you will start to see, cells from the urine or mesenchymal stem cells are also very common somatic sources for iPSC generation. And again, here, when you're thinking about what cell type you are trying to reprogram, there are certain considerations that we take into account, right? The first is how invasive is this method for obtaining the sample that you want, right? skin biopsies and blood draws are quite easy and non-invasive. So they're very, very common ways by which we can, you know, isolate cells and needed for reprogramming. The other is sample size or cell size, right? With certain reprogramming methods, the number of cells that you need is quite important. And again, we'll talk a little bit more about this in a second, but um, there are certain methods, for example, in urine-derived epithelial cells or mesenchymal cells that we can reprogram. The number of cells that we can obtain from that sample might be a little bit lower, right? And is that a consideration that you should be keeping in mind? One very, very quick example of this is when using blood-derived samples, right, or whole blood from pediatric patients. We can only get around 5 ml of blood from pediatric patients typically. And so taking that and after you do your PBMC isolation, where you're going to actually reprogram, you're going to have a smaller number of cells that you're able to work with. So is that something that, you want to consider before you even start reprogramming at all? The other consideration, which is maybe should have been at the top, really, is the ability of these cells to reprogram, right? For the most part, most proliferative cells tend to reprogram well, but there are certain cell types that don't reprogram as efficiently. One very clear example of this is as erythrocytes from your blood, right? Your red blood cells. They're non-nucleated. They don't reprogram very well, right? And again, we talked a little bit about this, but upfront manipulation requirements, the reprogramming method itself, and then your downstream applications of your iPSCs. This is very, very important, and I think there is some really, really nice there's some really, really nice evidence in the literature to indicate that once you reprogram a somatic cell into an induced pluripotent stem cell, that induced pluripotent stem cell maintains a tiny bit of memory that allows it to differentiate back into the original somatic cell source a little bit easier, right? So again, downstream applications might be another consideration of your cell source in the beginning. Any questions so far? I know that there's a couple in the chat. Let's see here. Can you elaborate on the rationale need to differentiate hematopoietic cell genetics from mRNA-based reprogramming? Yes, I'm going to talk about this, Tyler, a little bit more, but basically for mRNA reprogramming methods, the protocols that we have and the kits that are commercially available typically require an adherent cell type to reprogram with mRNAs because you're going to be delivering your mRNA into these cells, right? And blood-derived cells or PBMCs are cultured in suspension. So it's a little bit harder to do that when the cells are floating around as opposed to be attached onto the plate. And that's why we typically need to differentiate them into endothelial progenitors to make them into a monolayer culture of cells that are proliferative and then start our reprogramming.
Hey, thanks,
Um,
Omar. Could I actually follow up on that?
yes, please.
This is Tyler. So
Yeah, yeah, go ahead, Tyler.
we've been doing Sendai-based reprogramming of T cells. So I guess the question is kind of how would Sendai differ, or how would the RNA differ from the Sendai method? As you said, the T cells or PBMCs aren't adherent. They do, I guess, what people sometimes describe as loosely adhere to the bottom. So they are, if minimal disruption or minimal manipulation, they kind of stay on the bottom.
True.
So definitely harder to work with, but we have been able to make it happen with Sendai at least.
It does, yeah. We actually, and I'll talk a little bit more about this. We have protocols specifically for T cells as well. And this is primarily because of the mode of delivery, right? So Sendai or viral delivery is a little bit more efficient in this case. And Sendai specifically allows you to transduce a lot of different cell types quite easily. So you are able to do that in a suspension-based workflow. You'll notice that for the most part, we call, at least in our Sendai kit, we call that part of the step a spinfection, where we do the transduction while we're spinning the cells as well to get them to kind of pellet down a little bit more. But there are certain techniques that can be employed here, and we can talk about this a little bit more later on, and I'm happy to kind of go over this one-on-one with you, Tyler, to give you more specific recommendations too.
Great, thanks.
Of course. Um, yes, and I see Angel said the same thing, right? PBMCs are resistant to mRNA-based reprogramming until you basically get those adherent cells that will be more easily transfected with mRNA-based methods. So we're going to do a slightly deeper dive into some of the reprogramming kits that are currently available. To be completely honest, and just to call this out, Tom and I do work for Thermo, so our expertise are going to be slightly more lean on some of our offerings too, but you'll see that we're going to talk about different methods and the principles that we're going to talk about do apply globally as well. So at Thermo Fisher, we have a kit called the Epi5 Episomal iPSC Reprogramming Kit. And if you remember from that very clumsy slide, this was one of the methods by which you can use episomal vectors or plasmids that have your transcription factors or your reprogramming factors cloned in so that you're able to deliver this plasmid to the cells and have the cells start to express your protein of interest. Again, in this case, it's so that these reprogramming factors can be expressed in the cell. And in this particular kit, you'll see that we have the reprogramming factors in 5 different plasmids that we deliver. You have an OCT4 over here, a SOX2, and a KLF4 on this vector. You have CMYC on this vector, but then you have a bunch of other interesting genes that you see over here. So Lin28 and a dominant negative form of P53. And so, although these two genes weren't named in the original reprogramming paper, since then, right? This was the reprogramming first reprogramming paper came out in 2006 from Shinya's group, and so since then there's been some work that was done to try to better optimize those reprogramming efficiencies because at the time, right, although we could reprogram, the efficiency was really low, like less than 0.01%, right? And people figured out that there are certain other transgenes that you can add to enhance that reprogramming efficiency. These are 2 of them, right? So Lin28 and a dominant negative form of P53 allows you to improve reprogramming efficiency in this case. And then you'll notice here that almost all these vectors have an EBNA1 construct as well. So the way this works is when we deliver these vectors either by electroporation or lipid transfection into our somatic cell, right? These plasmids are going to bind to the chromosomes via the EBNA1 protein that's encoded in the plasmids. And this will allow then the expression and translation of your target gene specifically, mainly being those reprogramming factors that we talked about. And as such, it's really, really important to know that for the most part, these episomal vectors remain in the cytoplasm, but there has been in the literature some call-outs to say that there is possible integration of these plasmids via recombinating, right, in certain parts of the genome. Although that's a lower likelihood, it still has been noted. And eventually, right, once these proteins get expressed, you start your reprogramming cascade. But those episomal vectors do not remain in the cells forever, right? As these cells continue to divide and propagate over many, generations or passages, these episomal vectors are usually lost at about a 5% rate per cell division. And this is due to a bunch of different things, one being errors in DNA replication, right? But also partitioning. So if you imagine these five plasmids in one cell, when that one cell divides, right, their distribution between the daughter cells may not be five and five, right? You may get a partitioning effect where one daughter cell may get more of those vectors than the other. So that's another way by which these episomal vectors are lost um, over time in iPSC cultures once they're established. So this basically is the basics of the episomal method to reprogram. But what about the Sendai virus method, right? So just as a bit of a background, the Sendai virus is actually a respiratory mouse parainfluenza type 1 virus that is noted in rodents, right? It is an RNA-based virus. It replicates exclusively in the cytoplasm. It's non-pathogenic to humans, which is pretty important, but it also has pretty much a very, very low possibility of altering the host genome, i.e., it's one of those non-integrative methods, right? And with the Sendai, you'll see that a lot of folks that use the Sendai will know the term cytotune. This is just a name of a product that we actually offer at Thermo Fisher, where we've packaged those reprogramming factors into a Sendai virus particles, but we've also slightly altered the viral backbone, right? So what we did was we deleted the fusion gene or the F gene in the viral genome here, which is responsible for producing more infectious virus particles, right? So once the virus transduces a cell or infects a cell, delivers its payload and can start, expressing the transgenes. Typically, with normal viruses, the viral genome gets repackaged and then released so that the viral particles can infect surrounding cells, right? So this repackaging process is dictated by this fusion gene. And so by deleting it, we stop any further transfection or subsequent transduction, excuse me, of cells after we initially administer it, right? As I mentioned, the cytotune vectors or the Sendai vectors in this case can transduce a wide range of different cell types, and this is because they use the sialic acid receptor as their main mode of transduction, right? Again, allowing you just a little bit more freedom in terms of what original somatic cell type you want to reprogram. And actually, you can have really high transduction efficiencies at low multiplicity of infections. And multiplicity of infection, or MOI, is basically just the ratio of viral particles to effector cells that to target cells that you're looking to transduce, right? So how many viral particles am I intending for each cell, basically? And so that was a little bit of a background on the Sendai reprogramming. But if we talk specifically about the kit, you'll see that at Thermo Fisher we have the Cytotune 2.0 reprogramming kit, and it comes in three tubes like this that are shipped on ice and sorry, they're shipped frozen and should be kept frozen at -80 until you're ready to use them, right? And those three tubes, each of them has Sendai viral particles that are loaded with different vectors, right? So the first tube or Sendai particles have the KOS vector or the KLF4, OCT4, and SOX2 genes, right? The second tube has the c-Myc vector, and then there's another tube that has an additional KLF4 vector. Now, this may be a little bit counterintuitive as to why would you have two different transgenes for KLF4. And to that end, I'll say that one very important aspect of reprogramming when you consider the four transcription factors is gene expression, their relative gene expression to each other. So again, similar to that Lin 28 and dominant P53 modifications that were done to the episomal vectors to improve reprogramming, we found out that the relative expression patterns of those four transgenes can also dictate the efficiency of reprogramming. So by including an additional KLF4 vector, we see that we can increase that reprogramming efficiency as well. Where you can see here, compared to the legacy cytotune kits, the more optimized polycistronic KOS4 vector with an additional KLF vector as well allows for better efficiency and better
Hey,
reprogramming of
it's
both
almost here. You don't
fibroblasts
want to
as
get
well
out in the
as
warm?
blood-derived cells. Okay, any questions so far? Let's see here. Are there any chances of viral genome integration into somatic cell genome, like you said in the case of few episomal vectors? I would say there always is a possibility that something goes wrong, but the chances of that happening with the Sendai is quite low because it does replicate in the cytoplasm and it is an RNA-based virus, right? So that would be a slightly lower. Then your cytotune vectors also have a safety feature to, no, absolutely correct. I'll go back over here. So for two of those vectors, we have built in a mutation in the promoter that allows you to clear those vectors faster by culturing your cells at a slightly higher temperature. We're going to talk about this later on as well too, Angel, but you'll see that similar to what we talked about in terms of episomal clearing from the cells as time passes, we also don't want viral particles in our PSC cultures over time, right? So we look to generate a viral-free PSC line by the end of this protocol as well. And one way to do it is to get the cells to clear the virus by themselves. But we have built in, these heat mutations or heat-sensitive components into our vectors so that we are able to clear those viral particles, or at least two of them, slightly faster than you would normally as well. So good call out. Excellent. So now that you kind of have an understanding of the basics of reprogramming, right? What we're really trying to do here. One thing I wanted to call out, just in terms of nomenclature that we're going to see over and over again, whether on the reprogramming side or on the PSC culture side, is a nomenclature that describes the way in which we culture these cells. Whether we have these PSCs on a feeder-based or feeder-dependent culture system, or in a feeder-free culture system. And I'll talk about this again. We'll talk about this more in subsequent lectures when we go over the PSC culture side. But even on the reprogramming side, there are ways in which you can reprogram your iPSCs and maintain them as feeder-dependent cultures, where your iPSC colonies that you see over here are actually sitting on top of a layer of heat-inactivated mouse embryonic fibroblasts that are in the bottom. Or we can remove those MEFs, as we call them, or those mouse embryonic fibroblasts, and replace them with a defined matrix or ECM, so that we can support the attachment of these induced pluripotent stem cells. Because iPSCs do not like to sit on plastic. And again, don't worry about this. We're going to talk more about it later on. But just wanted to introduce this nomenclature, right? Feeder dependent cultures and feeder-free cultures. We're going to mainly talk about feeder-free cultures for the most part. The idea from moving away from these feeder-dependent cultures is we didn't want a culture system that had both human iPSCs as well as mouse embryonic fibroblasts. So you have two different species in one dish, which makes it a little bit more complicated to analyze your results downstream. But also, the feeder dependent cultures tend to take a little bit more prep time. You have to prepare your mouse embryonic fibroblasts, plate them before your iPSCs. And so just to maintain the cultures as only human cells, we most folks you'll see have moved into a feeder-free PSC culture system. But this isn't really to say that one culture is better than the other. They both have certain advantages. Feeder-dependent cultures tend to be really robust as well, so there are definitely applications for both. So for the reprogramming, I promise that we're going to do a slighter deeper dive into the cytotune Sendai reprogramming protocol slightly. So here I'm showing you the schematic of the reprogramming protocol for fibroblasts, right? That takes anywhere between 3 to 4 weeks usually. And the way we start our reprogramming experiment here is we plate our fibroblasts in fibroblast media. Typically, you'll see this designation of day -2, and because day 0 is when we're going to actually add the viral particles, right, the Sendai particles, and start this whole process. But you'll see that 2 days before, we usually 2 or 3 days before, we plate our fibroblasts in fibroblast media and allow them to grow to anywhere between 40 to 80% confluency, at which we then call time 0 is when we're performing the transduction. This is when we're going to be adding those cytotune reprogramming vectors to our cells to start this whole reprogramming cascade. From that point on, from about day 0 to day 3, you should notice a shift in morphology of your fibroblasts. So they're going to start turning a little bit more epithelial in nature, and they're going to continue to grow for about a week. Right at that point, slightly changing in morphology, and I'll show you a video in a little bit. It's quite clear when they do. But around day 7, we typically will dissociate these cells and then replate them back down. But now we're going to actually plate them down onto a very particular matrix because at this point, the cells are pretty much ready to start for a lack of a better expression, showing themselves as pluripotent stem cells, right? So in this feeder-free method, we actually plate them back down onto vitronectin-coated or Geltrex-coated or Matrigel-coated or laminin-coated plastics, right? If we were doing the feeder-dependent method instead of the matrix here, we would plate these cells back down onto those heat-inactivated fibers that I showed you. But again, for simplicity purposes, we're going to be talking about matrices here. So we're going to plate the cells back down onto a matrix on day 7. And and then you'll see that a day later, we will switch media. So we will no longer be feeding these cells with the fibroblast media. We're going to actually change to a media that is more suitable for these pluripotent stem cells, right? In this case, is Essential 8 media. That's a little bit lean, but it's shown probably one of the best reprogramming efficiencies as well in terms of media support here. And from this point on, from day 8, actually day 9 to day 11, you will start to see the emergence of these colonies, right? And they're going to be very, very distinct in morphology. And we're going to talk about that. These will continue to grow over, 2 to 3 weeks. And around day 19 to day 20 of that protocol is when we will typically harvest these cells and passage them over from the mother plate into a subsequent plate. Now, again, don't worry, we're going to talk about all of this. But the one thing I really wanted to go over before is sort of talking a little bit about the MOI or multiplicity of infection, right? On day 0, I mentioned that we have to add our virus, right? Our viral particle. But we all know that the ratio of viral particles to the target cells is important, right? So for those 3 vectors within the cytotune kit, right, in those 3 tubes, we do have some recommendations on the multiplicity of infection, or the MOI, where typically we want 5 KOS viral particles per target cell, 5 c-MYC particles per target cell, and 3 additional KLF4 particles per cell, right? But knowing that, right, how do we figure out from the volume of from the tube how much to add in terms of volume, right? Because in the kit, these 3 vectors are supplied as in liquid format. So the easiest way to do this is to take your recommended MOI, in this case, our 553, and we're going to use this particular equation to calculate the amount of virus we're going to be adding for each of those 3 vectors, right? So let's take the first one, for example. We have the KOS vector that has a recommended MOI of 5, right? So 5 referring to this vector right here. We're going to multiply it by the number of cells that we count on day 0. And then we're going to divide it by the titer of the virus that you can find in the certificate of analysis that you'll receive with your kit itself, right? And so based on that, we know, in this example, we have to add 10 microliters of the KOS vector into our media. And then we repeat this for each of the other vectors, making sure to add, the correct amount of volume from each of those vectors to the same cells, right? Because you want the same cell to be exposed to all 3 vectors in order to initiate reprogramming. Now, I kind of just threw this at you and I said, well, the recommended MOI is 5 for the KOS vector, right? But what if we did not know that? We know that because we've done a lot of background work. But what if you're working with a newer cell type, right? Not fibroblasts, or a really particularly hard to transfect cell. Well, there are some other tools that can help you figure this out. We have another kit that I'm showing here that is basically can be used as a control, right? Before you start your reprogramming experiment and spend a lot of time optimizing how much of that virus you want to give to each cell to properly transduce it, and at the same time probably spend a lot of money. What you can do is you can employ some of the controls that we have to actually figure out how much volume you would need with a Sendai virus to transfect your cell type, particularly. In this case, there is a Cytotune GFP Sendai reporter. This is a viral particle, a Sendai viral particle that was loaded with a GFP construct, right? So not the KOS vector, not the KLF4 vector, not the c-MYC vector. This is completely different. But what this allows you to do is basically add in the Sendai viral particles and figure out how much of those do you need to add in order to get proper transduction of your particular cell type, whether you're working with fibroblasts or CD34 hematopoietic stem cells, T cells. It can be a little bit of an easier way to do this, right? Now, one thing I have seen folks do is use similar methods in order to also indicate that the cells have been transduced, right? So they would use this GFP Sendai reporter. At the same time, they would also use the Cytotune Sendai reprogramming kit vectors. And I would caution away from that, or recommend that not be done, because Sendai viral particles will compete over that I can't say that word. The receptor basically that they bind to, they will compete for it. So adding a Sendai virus that's not going to actually do the reprogramming is going to probably lower your efficiencies there. So during this whole process, right, I talked about adding that virus on day 0, right? And on day 0, what you want basically for fibroblasts specifically here is to have a confluency of around 80 to 90% on the day of transduction. This could vary quite a bit from one cell line or one patient sample to another. So just be cognizant of that. Those that days before transduction, I've showed you in that schematic were day -2, but for certain patients, that could be maybe a day -3 or day -1, depending on how fast these cells grow. But on day two, right, I briefly described that those cells, as they start undergoing that reprogramming cascade, will shift from their fibroblasts, at least, will shift from their mesenchymal morphology to a more epithelial-like morphology, right, as they undergo MET. And this is a little bit more evident on day 7 as well, as you can see over here. Now, on day 7, we replate those cells back down either onto coated plates or on MEFs. But around day 10, and we start culturing them in PSC media specifically, right? So around day 9 to day 11, you should start to see the emergence of these very distinct colonies that will continue to grow, right? These hypothetically will have emerged from one cell that I've reprogrammed and has continued to proliferate. And typically by day 19 to day 21, these colonies will get big enough and their morphology will be recognizable enough for you to actually pick them out as pluripotent stem cells and different from these other cell types that surrounding cell types that have not reprogrammed, right? And this is one very, very important call out, and I'll make it again in a little bit, but not every single cell is going to reprogram. In fact, the reprogramming efficiencies I mentioned are quite low, relatively speaking, right? I think the most we can get up to is 2 to 3%, which some of you may be thinking this is really, really bad. You will have a 3% efficiency, but just remember that originally we were at less than 0.01%, right? So going up to 3% is orders of magnitude higher that we've managed to improve on that over the years. In addition to that, right, not every single cell will reprogram, but there's also some cells that will also arrest midway through. They'll basically start that reprogramming cascade and maybe start to form a colony that looks something like this, but then stop growing or de-differentiate back. And that is common as well. So I showed you some pictures, but I'll try to play a little video for you on how this whole process goes, right? So this is a time-lapse video in one of our incusites, or one of the cultures in an incusite where we're basically imaging the cells as they're growing. And we'll start from day 0 over here, where the sendai viral particles are being added, right? And you'll start to see that by day 3, the cells will be more confluent, but this you start noticing that shift in morphology to a more epithelial nature. And the cells, again, will continue to grow and continue to reprogram. And at day 7, this cut is when the cells were passaged onto the coated plates, right? And you'll see here again by day 9 to day 10, these distinct colonies start to grow, right? They're very different in morphology. They're not those mesenchymal cells that we started with, but now they're these cluster of cells, if you will, or colonies as we call them, that will continue to grow over the next 2 weeks or so in that PSC media up until they're ready to be passaged or transferred over into the subsequent plate for us to keep growing them. Sweet. And I mentioned very quickly that the Sendai virus can be used to transduce a whole bunch of different cell types. There is a repository, and if you search PubMed, you can find a lot of different papers on using the Sendai virus specifically to reprogram different cell types. Interestingly, there's also folks that have used the Sendai Cytotune kit to reprogram a bunch of different really, really cool animals. Jean Loring was working with the San Diego Zoo. If any of you have been down there to help with the reprogramming of a bunch of different animals, including the white rhino that was, there was only 1 or 2 left in the world. Jean, I remember, also talked about reprogramming giraffe or elephant cells at some point. Just really, really cool, right? I've also seen folks in Canada reprogram feline and canine cell types. So there's a lot that you can do with these reprogramming factors for multiple cells, but also multiple species too. Any questions so far before I move on to blood-derived cell sources for reprogramming? Anything about the fibroblast reprogramming protocol specifically?
Omar, there was a couple of questions in the chat which I'm addressing, but
Yeah.
if you have any other thoughts to add, feel free to do so.
Let's see over here.
And I can read them to you if you'd like, but
Yeah, no, I could see them. How do you manually pick? I love all these questions, and we're gonna get to it. Don't worry. So I have that towards the end about growing the colonies, isolating, needle size, things like that. I will cover a lot of this in several slides later. How do you separate program cells from surrounding cells? Yes, we're gonna talk about this. Yeah, absolutely. We're gonna talk about it. Don't worry. I love it, but you guys are ahead of me a little bit. And I should warn you, we will be together for an additional, well, including this week, 6 weeks. But and Tom will Tom will tell you this as well. I talk a lot, so please excuse me. This is a formal apology from the beginning, but we'll definitely cover a lot of this too as well. So don't worry. Okay, so for blood-derived cell sources, the protocol that I've been showing you so far with the cytotune has been to reprogram fibroblasts, but the protocol is slightly different for different cell types, right? I mentioned the other very common cell source to reprogram is blood-derived cells that can be taken and isolated from whole blood, right? Ideally, there's a lot of cells in whole blood draws that don't reprogram very well. We talked slightly about this, right? Your erythrocytes that basically don't have a nucleus aren't cells that you want to reprogram. So typically, what you see folks do is after doing a blood draw, or taking or leukapheresis product, basically like a bag of blood, right? They're going to isolate a particular type of cell from whole blood in order to reprogram. The most common one that you'll see is PBMCs or peripheral blood mononuclear cells, right? These are cells that include lymphocytes like T cells, B cells, or NK cells, but also monocytes like macrophages and dendritic cells. And um, the way to basically get your PBMC population is you take your whole blood and you add it to a density gradient. In this case, either histoplaque or Ficol, and then you spin this down where you can actually see that the cells will separate based on their different densities. You're gonna have the plasma on top. Then your buffy coat is going to actually have the PBMC population that you want. There's going to be your ficol, your histoplaque, and then towards the bottom, you're going to have your RBCs or your red blood cells as well. And this is just one particular method to do it, right? Some folks can take whole blood and use different positive selection tools like magnetic beads to pull out cells like CD34 positive cells that are your hematopoietic stem cell population. Although they occur at a very low rate in normal blood, they are still detectable and tend to reprogram really well. You can also do similar things with T cells, where you're going to take your whole blood and select for markers of T cells like CD3, CD4, CD8, or even potentially even use negative selection markers, right? Where you're eliminating other cell types by pulling those out as well. And then I mentioned that from whole blood, if you are thinking about a particular, mRNA, for example, method that doesn't easily support the reprogramming of suspension-based cells, you can derive, endothelial progenitors as well in a monolayer in 2D, and then use those, for your reprogramming as well. But once you have your PBMCs, right, you can start your reprogramming from that point. And again, the Cytotune kit has a bunch of different protocols for these cell types that are outlined. So we're gonna briefly go over this one as well. I won't go into it into this one as much detail, but I wanted to point out a couple different things that we've learned along the way, right? So in the fibroblast protocol, I should put my pointer here, I'm sorry. In the fibroblast protocol, I talked about those day -2 to day 0, right? And this is a growth period where you allow your somatic cell to grow to reach the right confluency before you start your reprogramming on day 0. For PBMCs, this tends to take a little bit longer. So we culture our PBMCs, which we tend to culture in suspension, right? With a PBMC specific media that is usually supplemented by growth factors like SCF, GMCF, and IL-3 as well. And one thing I will note here is we have seen, and there has been some data in the literature to indicate that this expansion period or culture period prior to transduction for PBMC specifically, the longer it is, the slightly better the reprogramming efficiencies tend to be as well. So another thing to keep in mind is this period prior to starting your reprogramming, having that be slightly prolonged allows the cells to slightly adapt better to a culture system, especially if you're using frozen samples to start, right? If you're taking PBMCs from a frozen stock. At this point, on day 0, we add our reprogramming cytotune vectors, and you'll see this protocol is slightly different, right? Because these cells are in suspension, for the first 3 days, you might actually start to see some cells that attach, but these cells aren't the ones, like Tyler was saying, these aren't the cells that are going to be the ones that reprogram very well. So we typically leave those behind, and we take the suspension-based cells, and only on day 3 do we actually start to plate those cells down onto a matrix-coated plastic and start to shift again, moving away from the PBMC media. Now we are going to move to a media that's specifically for those hematopoietic progenitors to help push those along because they reprogram pretty well. And then on day 7 or 8, just like before, transition into the PSC media to help support the emerging PSC colonies that are going to be coming up. Now, as a resource for you, I wanted to introduce this particular paper from Joe Wu's group. It was published a while back, but it does have a really nice comparison of the different reprogramming methods that are currently available for researchers across the board. They talk about, the possibility of integration, reprogramming efficiencies, cost, and effort or labor associated with it, with that particular workflow. So it's definitely a good resource and a good read. Another thing I wanted to briefly introduce, and we're going to talk about this towards the tail end of our lecture series, is reagents that are going to be very similar to the ones that we talked about with the Cytotune, but slightly different in the sense that they're made for specifically for clinical applications, right? We talked a lot about the Cytotune Sendai reprogramming kit, right? But I want you to know that sometimes when we want to take those cells into the clinic to use them for example, for therapy, the use of traditional products like the Sendai Cytotune 2.0 may not be enough, right? There is a lot of considerations that we have to take in terms of the materials that we're using and whether or not they're suitable for clinical or bedside applications, right? The FDA tends to have a lot of regulations around what products and what components you can have in your reagents that you're using with your cells, specifically for cell therapies, right? And one good example here is this cytotune kit. So the cytotune kit that we've been talking about is basically designated as a research use only product, but there is alternatives under our cell therapy systems group where we do have alternatives that allow you to use very, very similar products. In this case, this product is called the CTS Cytotune 2.1 Sendai Reprogramming Kit. It's pretty much exactly the same concept and idea as the RUO offering or the research use only offering. But the reason why I'm bringing this up is there is slight changes that are made to these fit for purpose clinical reagents, right? That allow you to reduce risk that is associated with going to the clinic. For example, with the Cytotune kit, we mentioned that one of the reprogramming factors was c-Myc, right? But in this case, because c-Myc is a proto-oncogene and can actually drive proliferations of cells quite fast, there is a fear of transformation, right? That these cells might just continue to proliferate like crazy. And with pluripotent stem cells, that is a concern because these cells, as we heard from Tom last week, these cells by nature can self-renew indefinitely, right? So we replaced the c-Myc vector in this 2.1 kit with an L-Myc vector that still does a similar function of driving proliferation, but it has a weaker transformative ability in this case. In this kit as well, the fit for purpose kit or the CTS kit, as I'll call it from now on, we removed BSA from viral dilution buffers. BSA is bovine serum albumin, right? This is an animal-derived product, and for clinical applications, the FDA likes to stay away from animal origin products and more into what we call a xeno-free workflow, which again we'll talk about in later lectures. But I wanted to introduce this concept that even though you could be doing great work with a certain reagent, it's really, really important to consider your endpoint, right? Where do I want to be going at the end of my work, right? If I'm working towards a clinical therapy, for example, taking those stem cells, deriving dopaminergic neurons that I can transplant back to help save Parkinson's patients, right? Or reduce their symptoms. If that is the goal, then I have to consider what reprogramming kit I'm using, right? The RUO kit is fit for research. The CTS kit is better suited for clinical applications. And again, very similar to the different protocols that we talked about, but there is recommendations on how to reprogram different cell types with the CTS kit, right? The clinical kit, if you will. Again, in what we call xeno-free workflows, right? Making sure that there are no animal origin components, whether in the kit or even sometimes in the different medias that you use, right? I mentioned blood-derived reprogramming. We can use a CTS version of the StemPro 34 media that is specifically geared towards the culture and support of hematopoietic stem cells or CD34 positive cells, right? In this case, we also have a CTS version of the Essential 8 media that you need to reprogram with as well. So keep those little things in mind, just in terms of what reagents you're using and what's best for your particular downstream applications. And a great point and great transition here. There is a lot of different cell types that both the 2.0 and 2.1 kit can reprogram, right? This is just several of them, including PBMCs, those CD34 positive cells, T cells, or fibroblasts. And you will notice that the 2.1 kit's efficiencies are slightly lower than that of the 2.0 that you see in the blue bars. And this is primarily due to those small alterations that we talked about, right? Making sure that we replace the c-Myc with an L-Myc, so these cells are less transformative, which makes them slightly less proliferative, and some will argue that that makes the efficiencies a little bit lower as well. I see there's a question in the chat. Is there a difference between reprogramming efficiencies in naive versus activated T cells? Uh, Good question. You definitely need to activate your T cells to start the reprogramming well. And I'll kind of go back to this particular figure. You'll see that we actually, in this case, when we're culturing the T cells prior to the day of introducing the Sendai vectors, we culture them with IL-2, but also with dynabeads that have CD3 and CD28 epitopes that allow us to activate those cells. And so that makes them more proliferative. And then because of their reprogramming efficiency or our ability to reprogram is slightly better. Great question. Excellent. Sweet. Now, we talked a lot, I would say, about the different reprogramming methods, the protocols, but what are the cell types, or what are other considerations that can affect your reprogramming process, if you will, right? And I would argue that that also includes the tail end of this protocol, right? We talked a lot about the beginning, right? What cells you're using, how long you're culturing them for, and what media. But towards the end of our protocols, we're switching to a PSC media, and for feeder-free reprogramming, we're switching to a some sort of matrix, right? That we're coating the plastics with. And um, in this case, we have done some internal work on our end on Thermo Fisher, and we've noticed that leaner medias tend to do better. Leaner PSC medias tend to do better in terms of reprogramming. So, for example, Essential 8, and we'll talk about this, I think, next week. Essential 8, as the name suggests, has 8 essential factors that is basically the bare minimum to keep PSC cultures afloat so that they don't differentiate or die, right? And that lean media system tends to do pretty well in terms of reprogramming compared to other maybe more robust offerings like StemFlex or MT01, for example. Now, the other thing I wanted to call out is the matrix itself is also important and can dictate the efficiency of reprogramming, right? So at this point, you'll see that in those schematics, the cells on day 7 or on day 3 were replated onto vitronectin-coated plates to kind of continue driving the reprogramming process, right? But there's other matrices that are available that you can coat the plastic with that can actually give you better efficiencies or improve your ability to derive a cell line at the end of that 3 to 4 week period. PSCs really love laminins, right? So using a laminin-based matrix, whether it's laminin 521 or laminin 511, after you replate the cells, tend to do really well at getting those cells to stick down and attach. But there's also Geltrex, Matrigel, and Vitronectin that also work really well. And mind you, Geltrex and Matrigel are both membrane extracts, right? Basal membrane extracts. So they contain a whole bunch of different proteins, including laminins, but also collagens, fibronectins, integrins, and many others as well. Excellent. We have, I think, one more question in the chat about how do you determine reprogramming efficiency? Is it initial colony formation or cells that go on to produce a clonal iPSC cell line? Typically, the easiest way people will do it, Angel, is that they will count the number of colonies. And knowing the original starting cell number that we used for our transduction, you can get a relative idea of the reprogramming efficiency as well. So based on how many cells you use and how many colonies you end up with, given the fact that there is an assumption here, right, that the every colony generates from a single cell, then you could basically divide those two numbers by each other and get a reprogramming efficiency. Excellent. And okay, so we talked about episomal, we talked about the Sendai, but there is another really, really interesting method of using mRNA to reprogram that I think we should definitely talk about, right? This is not something that we offer at Thermo Fisher, but I wanted to also make sure that we cover this because it is more of a goal in the field, right? If you remember those two factors that we rated all of the reprogramming methods around safety and efficiency, mRNA would be at the top, right? Very safe. We're not using viruses or episomal vectors, but also can be quite efficient when employed correctly, right? So with mRNA reprogramming, there was a first generation reprogramming kit that was launched a while back that basically included five mRNAs encoding the reprogramming factors plus Lin28, right? But the reprogramming protocol initially in that first generation kit was very, very cumbersome. If I can get this up. This is a schematic of that protocol where in order to maintain the expression of these mRNAs, because mRNA have a short half-life, right? The user was required to do 12 to 16 daily transfections, where you're resupplying the cells with those five mRNAs um, every single day for 16 days, right? And in addition to that, you also had to apply B18R protein to neutralize the interferon effect because cells normally, when they see foreign mRNA floating around, they have a defense mechanism, and so you had to block that as well, right? So as you can see, that's quite cumbersome, very, very workload heavy, but also um, might be a little bit damaging to the cells, right? Transfection is not an easy process, and doing that 16 times in a row might damage the integrity of your cells as well. So that was a big downside. But on the bright side here is there was no expression of transgenes within days of the last transfection. So you could clear your cells of any transgenes that you use in this process rather quickly, which typically takes a much longer time with either viruses or episomal vectors. But this method was mainly again compatible with adherent cells, primarily because of your transfection-based methods. And again, I should mention here, in this case, they were using RNAi Max as a transfection tool, right? Instead of lipofectomy. Several years later, though, there was an improvement that was made on that original reprogramming protocol and kit, where instead of five mRNAs, the third generation kit had six reprogramming factors. So very similar to the episomal factors that we described, or the transgenes in the episomal vectors we described. So OX4, SOX2, KLF4, CMYC, and then Lin28, and then in this case, we're also adding NANOG, another PSC specific transcription factor. Now, along with those six mRNAs, there's also microRNAs and mRNA encoding for immune evasion factors like E3, K3, and that B18R that we talked about, right? But with this particular kit, you didn't have to do the 16 daily transfections. You only needed to do four to six, depending on the cell type that you're working with. And again, after the last transfection, you wouldn't see the expression of those transgenes anymore, which is desirable. But again, this method is mainly compatible with adherent cell lines because you are using some form of lipid transfection to get those mRNAs into the cells. Okay, so now that we kind of know the idea behind reprogramming, we can get an idea of what that process looks like, how long it takes, what we should be thinking about. At the end of that, the idea is to have a PSC colony that then you can continue to propagate, right? And I briefly alluded to this, but there is very, very specific morphological features to pluripotent stem cells that we can pick out just by phase contrast microscopy alone that I think everybody should be aware of. And we will continue to talk about this more as we continue to talk about how to culture these cells because it's super, super important. But I wanted to highlight some of those morphological features. So this is just an embryonic stem cell line, H9s as they're called, and they're cultured on mouse embryonic fibroblasts in this case. So this is a feeder-dependent culture. And I wanted to just show you what these cells look like, right? So the PSCs themselves are usually very, very compact. These cells love to be in very, very close contact with each other. So the closer knit they are, the better in this case. You'll also notice that these cells tend to have, or these colonies tend to have a very refractive and defined edge. You'll notice that it almost looks like you're shining a light from behind the colony, right? So the edges are a little bit brighter, and that's a distinct morphological features of those cultures. But if you zoom in and you look at individual cells, you'll also notice that those cells have a pretty large nucleus compared to their total cell body, right? So we usually call this a high nucleus to cytoplasm ratio, meaning that the majority of the cell is nucleus dominant. And inside of that cell, if you zoom in even further, you'll notice that there's also very prominent nucleoli, sometimes multiple. And again, that's normal with these cells as well. So once you kind of have an idea of what you're looking at, right, morphologically, this is especially in the context of reprogramming, right? After you've seen these colonies emerge, you could identify that they look decent and they look good. What do you do next, right? And there was a couple of questions about that in the chat. So the first thing to do is to kind of identify the PSC colony via morphology, right? And typically what we do is we'll go in and we'll circle the bottom of the well where we see that colony so that we can easily identify where it is in the plate, right? We can do that with a marker, but there's actually also some really, really nifty tools and microscopes that have these like little circles that go onto the actual objective and can be used to stamp the bottom of the plate as well as you're working your way through. The second thing that I would do is, and some folks don't do this as well, but depending on how comfortable they are with their ability to assess morphology, but you can employ live stains, right? Where you can stain these cells and these cultures for markers of pluripotent cells, like TRA160 that you see here in the red. And then also live markers like CD44, which in this case, in green, it stains the fibroblast cells. So with this method, you're very, very able to easily identify which cells are pluripotent and which are not, right? Aside from just looking at morphology. The third thing that we usually typically do is that we manually score the selected colonies that we have identified as good, and we wanted to continue on with usually some sort of needle or a pipette. I typically like anything between a 22 to a 25 gauge needle, and we typically start by, like I said, identifying that colony. And then we take this needle or pipette and we make these horizontal lines across the that particular colony that we want to transfer over or we want to manually passage, right? We then rotate the plate 90 degrees and make those same, little scores or cuts so that we have this grid pattern that we form over, cutting our colony basically into these tiny squares. And I should mention here that um, how big those squares are matters, right? So it takes a little bit of practice, but if you wanted to, you can even do it just on regular plastic without cells, just to get comfortable with the motion. But you can kind of think about it as making sheets of cells, right? In those small squares. And the idea is that once I've cut these cells into small sheets, I can then transfer them over as clumps, right, and have them be propagated in the next plate, right? But if that sheet or that square is too big, it's going to fold in on itself once you transfer it over, and it's going to be messy, and those cells might start to differentiate as a result of that. If the on the other hand, if that square or that sheet is too small, then those cells are going to be very, very stressed out, right? I alluded to this before, but stem cells really like to stay close to each other. They do not like to be passaged as single cells. So doing it this way, we avoid or we try to reduce the stress on the cells. Once we have that, then we typically then go in with a sterile pipette. Usually, I like a 200 microliter tip pipette, and I transfer over these squares that I've cut by gently scraping them over and pipetting them onto the into the new plate that has been already coated with our matrix as well.
Omar,
So
there was one question, hand raised from Noreen.
yes,
Hi. So
yeah.
quick
Sorry, Neri, go
question.
ahead.
Yeah, no, it's okay. So in terms of the college, like the when they're reprogrammed, sometimes they're close to each other and they merge, right? So
Mhm.
is, separating and scraping and making that colony number one, how much of a issue that might be towards the end that it's two different reprogrammed cells that actually, form the
That's
one
a
colony?
That's a great question. So ideally, what you want to do is you want to create clones, like separate clones. So if you've seen that two colonies are merging, I would tend to pick colonies that are more on their own
Mhm.
and further away from each other. But if that is not an option, and there's two colonies merging, and you see one of them potentially is a little bit bigger or looks slightly better than the other one morphologically, is you can go ahead and scrape a clear border around the cells that you want to pick, right? And this
Right.
is primarily why maybe researchers like myself back in the day would use these needles because we would have a lot more control about where we were cutting or scoring, and we wouldn't want, for example, these types of cells, right? So I would only
Right.
make squares on the cells that I want to lift specifically. But I agree with you. I think the two colonies together, usually not the best approach unless it's kind of a final resort. That being said, it's not the end of the world because you can further clone out later on. But from a starting point, it's typically better to start with more defined single colonies.
Yeah, thanks.
Of course. Would you use the same needle, score different colonies, or use a new needle for every one? Um, I guess it depends. I honestly wouldn't. If it's from the same plate, I can cut with the same needle. But if I find that the needle is somewhat damaged, or I'm scared that I might be transferring some other cells in the process, then I'll use a different needle as well. But typically, I would use the same one. Okay. So talked slightly about how to make those cuts right manually, and this was maybe a little bit more of a traditional method that honestly I still like and I employ. Catherine, Kita, I don't know if you guys still do this in the lab as well in terms of manual scoring, but I find that I have really good control with it. But there's other alternative methods, and some of them are actually starting to become really, really nice, right? So um, uh, it was either at my time it was either that needle or if we were strapped a little bit, we would take a glass Pasteur pipette and we put it under a Bunsen burner and then pull the pipette, which makes it really, really thin. And we can get this almost knife looking like pick, right? That we can also use as well. Then and because it was run over a Bunsen burner, it would be sterilized too. Uh, but those are more traditional methods. It's there is somewhat of a learning curve, which is why I mentioned if you wanted to, you can just take a a, a plastic plate and kind of just practice this technique. Because if you don't run the needle bevel down, for example, and run it at a particular speed, your hand will shake. So you're going to make squiggly lines instead of straight lines, which makes it harder to pick up those like little squares, right? That being said, there's tools like that are commercially sold as well. This is just one example of the Easy Passage tool. This tool is basically a roller that has a bunch of needles across the entire roller where you can typically just take the roller, put it on your plate, and then run it one time to make these first set of parallel lines, and then rotate your plate again and do it one more time to get the cross stitch or the cross hatch here, right? This does a really, really good job of maintaining a nice size for these squares or these sheets to make, but slightly at a disadvantage when you don't have as much control onto which cell types you are rolling over or not, right? Because you're not seeing them as easily. So this is one other kind of downfall. Right now, there's even much more advanced techniques, or even cooler, where people are using more automated methods in order to manually assess, not manually, sorry, automatically assess the morphology of the cultures by using, different algorithms. And now with the advent of AI, I'm sure that that will get better and better. But also specific automated tools to do automatic picking of the cells, where a machine, after deciding what the borders of the colony is, what the perfect size of clumps to make, would then go in and do all of that automatically as well for you. Excellent. That being said, and we talked slightly about this before, but the early stages after reprogramming, typically you'll see a lot of folks do what we call subclonal expansion, where instead of taking um, all the colonies that are growing in one well and digesting all of them at the same time and moving them over into a new well, Quincy passage, you actually do that at the colony level, where you take a single colony and you only passage that one onto a new well, and then potentially another one into another well, right? And you do that for several passages. Um, Now, the reason for doing this is subclonal expansion tends to give you better results when it comes to vector clearing, right? Either removing the virus, the viral cytotune vectors, or the plasmids as well. And this is one example with the cytotune vectors, where you'll see that for the subclonal expansion, if we're looking at the Sendai virus by qPCR, in 5 different clones across multiple passages after reprogramming, that we can see a dramatic drop in the relative levels of Sendai virus, right? Where by passage 8 to 10, we can no longer detect the viral backbone, right? And although this is somewhat true with the bulk expansion method, you still see a continued loss of the viral backbone of the viral vectors over time as you passage these cells after reprogramming, but it tends to be at a much slower rate, right? And the reason why I mention this is in stem cell research, we like to work with lower passage cultures. Uh, and the reason for that is these cells, as Tom mentioned last time, right? They can grow indefinitely, but they can also accumulate mutations over time. So working in lower passages, once they're characterized and viral vector free, right? Yeah, viral vector free, then we can actually start using these cells sooner for downstream applications, like the research that we're intending, right? So this is another consideration just to keep in mind. And very common characterization methods that you'll all see, probably all do in the lab, is just some form of staining, right? We talked about the live stains a little bit, but commonly staining your cultures and looking at different markers that are associated with pluripotent stem cells after reprogramming is also something that you'll see very, very often. And these markers include things like TRA160, that's a cell surface marker. SSEA4 is another cell surface marker, while OCT4 and SOX2 are two transcription factors. So there should be nuclear stains that should be found in your PSC population of cells. Again, this might be a little bit confusing, but if you remember, OCT4 and SOX2 were part of those reprogramming factors, right? And I just a couple of slides ago told you that we intend to clear those transgenes from the cells after a while, right? But the whole idea of reprogramming is that you deliver those transcription factors transiently initially, and then you allow the cells to endogenously start to express those markers on their own. So OCT4 and SOX2 are two markers that are seen in pluripotent stem cells in the embryo as well. And you can look at this multiple different ways, right? Looking at staining and immunocytochemistry. You can look at the growth rates of the cells and how well they're growing, they're reaching confluency. You can look at the cell surface markers by flow cytometric analysis as well. And the reason why I'm mentioning all of that, just in terms of uh, looking at your cells and different ways in which you can look at them, and we'll talk a little bit about this in the next several lectures, but it's really, really, really important to assess and characterize your PSC cells and cell lines as they're coming out of reprogramming, as well as during routine culture. It's well documented now in the literature that because of their proliferative capability, right, and their self-renewal ability, these cells can accumulate mutations. In fact, some of the numbers coming out looking at retroactive studies are quite staggering, with folks showing that around 20% of PSCs that have already been published have accumulated some sort of genetic abnormality. In particular, chromosomal gains and losses are very frequent, but then as well, subchromosomal copy number variants are also has also been detected, right? And um, there are several different ways about looking at your cells, from a molecular standpoint, making sure that the transcriptome and pluripotency is maintained. But on the flip side of that is also looking at karyotype of your cells, is also really, really important. This is typically done via traditional G banding methods, where you take live dividing cells, you fix them, and then you basically, in a drop, cause these cells to explode onto a slide, where then you can analyze the metaphase spread of the chromosomes under a microscope, and basically line, making sure that after our staining, that all of the chromosome 1 is there, and there's two copies of it, right? And this is the traditional G banding that we've seen in all biology textbooks, but there's really cool new spectral karyotyping um, uh, uh, methods that can also be employed here as well, that are a little bit more modern. But typically, the pros here in terms of using those methods is that they can be somewhat sensitive to different mosaicisms in the population of cells. So if you're looking at 100 cells and only, 5 to 10 of them are mutant and have some sort of abnormality, you can detect that. But on the negative side, if you will, these types of characterization or karyotyping tools tend to have a lower resolution. They can take about 3 to 4 weeks to before you get some data back, and they need a specialized person looking at that data to make that conclusion, right? And then it also does require live and dividing cells as well. We'll talk more potentially about molecular tools down the line, but I wanted to call this out again as another important consideration after reprogramming specifically is making sure your cells are one, karyotypically normal. Um, any downstream application that you use with these cells will really depend on whether or not they're indeed the genotype that you think they are or not, right? Plus the fact that you have to ensure that they're pluripotent, meaning that they can divide and give rise to all three germ layers as well. I won't go into too much detail about G banding specifically when it comes to karyotyping, but I did want to point out that this type of assessment allows you to look at, your chromosomal integrity, right? And it can detect things like chromosomal gains or losses, whether they're in specific small areas or smaller areas, or their entire gains and losses of chromosomes, like trisomy or monosomy. But there are other things that it cannot detect, right? So it can't detect single nucleotide polymorphisms or single nucleotide variants, because the resolution of those assays is not quite sensitive enough to go down to a nucleotide level, right? It cannot detect loss of heterozygosity, which has been reported to be somewhat of a problem with PSCs as well, right? Where you lose a one allele that was passed down from only one parent, for example. What it can do, which molecular tools cannot, is for the most part, is look at inversions and translocations, right? I go back one slide, because we're looking basically at a photograph, if you will, of those chromosomes. We can tell whether the position of a particular set of genes or loci have shifted, inverted, or translocated onto another chromosome too, as well. And then finally, I briefly mentioned the mosaicism part, but in those assays, and typically in all of cell culture, right? The goal, we kind of discussed this a little bit, the goal is to have a clonal population. But sometimes if a particular cell in your culture acquires a mutation that gives it a proliferative advantage, it can start to, grow in number and slightly take over that culture. So with the G bending karyotyping assays, you're able to detect cells that have a mosaic genetic mutation, right? Where some cells will be normal, as you see here, but some cells will have that mutation. And with G bending, you can detect that mosaicism down to 5 to 10%, which is quite low and quite sensitive too. And the reason why I'm mentioning really all of this is really just to kind of set up our next several talks about how we're going to continue talking about culture of these cells and downstream applications. But understanding the karyotype is really, really important. We've done a retroactive study looking at the karyotypes of samples that were submitted to our services group a while back, and we've seen that approximately a quarter of the samples that we've received have had at least one abnormality. And not only that, but those abnormalities also tended to have higher frequencies at certain locations in the chromosome. So aberrations or mutations in chromosome 1, chromosome 12, chromosome 20, and then the sex chromosomes was much more prevalent in that sample that we've analyzed compared to the other chromosomes as well, really indicating that there may be mutational hotspots, right, that we should be aware of. And same thing goes for pluripotency. When we looked at the global transcriptomic pattern of those same cells, we saw that a good portion of them were either a borderline in terms of their transcriptomic signature, not necessarily resembling differentiated cells, but also not resembling the pluripotent transcriptome. And this was done via two different assays, again with similar results. So again, just to really say that this is super, super important, that we have to make sure that our cells fit the definition that we want them, right? Pluripotent stem cells, by definition, are can self-renew, but then they can give rise to all three germ layers. And if they don't do those two things, then technically they're not pluripotent stem cells. And that can be dictated by their genomic background. So assessing their karyotype is also really important. And with that, I will get off my soapbox. And just as a bunch of resources for you, I put some links down to a free e-learning course around using the Cytotune kit, where you get to walk through step by step of each of the protocols for different cell types. Some of those protocols I've also linked in for the RUO version of the kit, as well as that clinical version I briefly mentioned. There's some really interesting and nice frequently asked questions that you can review as well if you're having any issues, as well as some videos that we've put together. And if you haven't read it, but this is a link to Shinya's noble acceptance speech, The Winding Road to Pluripotency, which is honestly a really good read. It kind of takes you into, gives you an idea of the thought process and how they came about this monumental discovery of, basically reversing back time for our cells, taking them from a fully differentiated somatic cell type and then turning them back to cells that resemble cells in our embryo states. Fascinating. But like I said, if you haven't read it, give it a read. It's a really, really interesting speech as well. And with that, I'll say thank you and happy to take any questions.
Omar, I'm going to make one quick plug here as well. Well done, that was amazing. But quick plug for the e-learning course that you mentioned here. I will say not only is it honestly a fantastic resource, completely free, but as humble as he is, Omar also completely built, designed, and generated this course online. So it's fantastic. It's a really great resource.
Thank you, Tom. You're too kind. But yeah, if you guys have any questions about that again, let us know. We're happy to help. And I believe next week we're going to start talking about the next part of the workflow, right? Which is after reprogramming, you now have an iPSC cell line, and then you can start culturing it. And we're going to go through all of those considerations about what it takes to culture these cells, different techniques, how do we passage them, how do we what applications we can use them for. And I think we're going to divide into two lectures, but we're going to also discuss 2D and 3D culture of those cells more recently as well. So I hope you're excited for that because I love that part. I'm biased, but I love that. Excellent. Any other questions in the chat I missed? Okay, we have Arjun has a question. Are there any major differences between embryonic PSCs and iPSCs besides how they are derived? And there are the major disadvantages to working with iPSCs over embryonic stem cells. Um, so they are different. Yes, they are. They do resemble slightly different populations. There is, for the most part, a consensus on, I would say, on the transcriptome that they're both relatively similar, but there are very specific genes that are very different. Also, with the iPS and the ES cell lines, there's also a difference in epigenetic modifications that are associated with either the embryo state or a reprogram state, which could be different. Um, I don't know if Catherine or Kita, you guys also have any other input here. I don't know whether it's easier or harder to work with ESCs or iPSCs. I would say it's very line dependent. There are some ES cell lines that are robust that I've been working with for a very long time, and there's some really easy iPS cell lines that I've also worked with that are amazing. But on both ends, there's also lines that I don't even want to think about anymore.
Yeah, I also think they are identical at this point, like for our purposes. So whatever is easiest to work with is best to work with, I think.
Yeah, clinical aspect is about the histocompatibility. So iPSC is matching with the donor, but ESL, we cannot. But there is another method to generate the universal iPSC or universal to cross the patient.
Mhm. And one other thing that I think I didn't want to mention too much of, but I think it is important is just in terms of the reprogramming, right? Originally with ES cultures, typically deriving them from the embryo caused the demise of the embryo, right? And this is a somewhat of a sensitive topic. It was for funding for a while for in the United States, right? So there was some oversight on how these things should be funded or whether or not they should be funded. Embryonic stem cell related research, right? And so with reprogramming, with the advent of reprogramming as well, we can circumvent some of those ethical issues that some people are concerned about with embryonic stem cell work too. Not to say that we completely eliminate them, but there is a definite progress there as well. So another thing to consider. Excellent. So if there's no more questions, we can stop right there. I told you guys I talked too much, right? I only had I want to say like 40 slides, but but it took me almost 2 hours to go through. I'm hoping I didn't bore you to death and that it's just me, Tom, Catherine, and Kitai left on the call because I can't see who else is on there. But if there's any questions, please let me know, let Tom know, and we're happy to help. And we'll see you next week.
Well
Perfect.
done, Omar.
Thanks,
Thank
everybody.
you.
Thank you, Omar.
Cheers, everyone. Have a nice week.